Organisms are constantly evolving. They adapt to different features of their environment, abiotic factors like temperature and pH, as well as biotic factors, or the other organisms around them. Interactions with other organisms can shape the evolutionary trajectories of the traits of an organism. We can represent the interactions among organisms sharing an environment as a network, where each organism is a node, and organisms that interact are connected by an edge. If we are specifically representing trophic interactions, where one organism preys on another and obtains energy from it, then the network is called a food web.
It's easy to imagine a food web of animals. The grasshopper eats the grass, the mouse eats the grasshopper, the snake eats the mouse, and the hawk eats the snake. But what about microbes? They exist in complex natural communities, and they participate in a wide variety of interactions, like cross-feeding and predation, but we don't have a good understanding of how these interaction networks function in nature, and how things like predatory interactions help shape these networks.
In this study, we addressed this question using 4 soil microbes: the nematode Caenorhabditis elegans as an apex predator, the bacterium Myxococcus xanthus as a mesopredator, and Escherichia coli and Flavobacterium johnsoniae as 2 basal prey species. Our main focus was the charismatic social bacterium Myxococcus xanthus. It is often used as a model system for studying cheating and cooperation because its complex life cycle includes several cooperative behaviors, such as social swarming and fruiting body formation. It is also a predator and has been shown to consume a range of both gram-negative and gram-positive species. However, it has not really been studied as a prey species.
There is one paper from 2011 published by Dahl et al. In the Journal of Bacteriology, where they look at the suitability of M. xanthus as a food source for C. elegans, but that's it. We wanted to better understand this relationship because both of these organisms are distributed at a global scale, and so we do think it's likely that they interact in nature. We wanted to understand what might happen when they meet, and in particular, how this might have influenced M. xanthus's evolution in its natural environment.
I developed a medium that would allow us to co-culture the 4 organisms, and my colleagues and I performed several experiments to investigate the interactions among them. First, we asked if C. elegans would act as a predator toward M. xanthus. We performed a binary choice assay where we offered C. elegans different patches of live and heat-killed M. xanthus. We used 2 strains of M. xanthus, a wild-type strain, which is motile on agar surfaces, and a non-swarming strain, in order to test whether M. xanthus motility plays a role in modulating the interaction with C. elegans.
Based on the results of the previous study, we were surprised to see that the majority of the worm populations actually left the plates, as you can see in panel A, where we show the fractions of the worm populations lost. In panel B, we show that of the worms that remained, it's a bit unclear whether they were making a distinct choice between patch A and patch B, the patch of living bacteria versus the patch of dead bacteria, although after 18 hours, it seems like there's a general trend toward them preferring the patch of dead bacteria.
In this assay, the patches of bacteria are small, and we can't distinguish between the scenario where the worms are actively avoiding the bacteria and the one where they assort randomly on the plate. So we did another assay where we inoculated live or dead M. xanthus onto half of a plate. This way we could see whether the worms expressed a preference to avoid or to interact with the bacteria.
We found that still, in this assay, the majority of the worms preferred to leave the plate, as you can see in panel C, although we did notice a statistically significant difference between the fraction of worms that stayed on the plate if there was only buffer available versus if there was any type of M. xanthus available, which indicates that if there is some M. xanthus available, it's more likely that some of the worms will stay on the plate. In panel D, you can see that in this assay, we got a better sense of any choice that the worms are making between interacting with M. xanthus versus not interacting with M. xanthus. So by 18 hours, the worms tended to prefer interacting with M. xanthus over not when the M. xanthus was dead. And by 42 hours, there seems to be a general trend of them preferring to interact with M. xanthus versus staying on the uninoculated side of the plate. Also, by this point, we still haven't seen any difference necessarily between the swarming M. xanthus and the non-swarming M. xanthus in terms of the way the worms respond to the bacteria.
Next, we tested how the presence of M. xanthus in a patch of basal prey might influence C. elegans' interaction with that prey patch. We did a binary choice assay like before, where the 2 patches that the worms could choose between were buffer, E. coli, or F. johnsoniae. Some of the patches had M-xanthus mixed in.
In this binary choice assay, we first allowed the worms to choose between a patch of F. johnsoniae and a patch of E. coli, in which case they chose the E. coli. We then let them choose between a patch of E. coli and a patch of E. coli with M-xanthus mixed in which case they chose the patch with no M-xanthus. We also saw them choose the patch with no M-xanthus when the choices were between F. johnsoniae and F. johnsoniae plus M-xanthus. When we let them choose between a patch of E. coli and a patch of F. johnsoniae plus M-xanthus, they clearly chose the E. coli patch. But when we let them choose between a patch of F. johnsoniae and a patch of E. coli plus M-xanthus, they started to prefer the F. johnsoniae, in which case the presence of M-xanthus in the E. coli patch actually changed the preference of C. elegans for the basal prey.
Finally, we set up a binary choice assay where both predators, C. elegans and M-xanthus, could choose between a patch of E. coli and a patch of F. johnsoniae. The only difference from the previous assays is that this time we inoculated M-xanthus between the two prey patches so that it would encounter them as it swarmed outward.
As before, the C. elegans clearly preferred not to interact with the M-xanthus. What you see here is the remnant of an E. coli patch, with M-xanthus coming in from the side in this sort of orange Pac-Man shape, and C. elegans worms clustering at a high density in the portion of the E. coli that M-xanthus has not yet reached. To test the response of M-xanthus to the worms, we calculated their swarming rate across the two different prey patches. In the F. johnsoniae patch, we saw that initially the M-xanthus swarmed quickly, but then the swarming rate slowed down. But this didn't change depending on whether there were worms present. However, in the E. coli patch, we saw that while M-xanthus tended to maintain a steady swarming rate throughout the course of the assay, after 5 days, they actually started swarming faster on plates where worms were present. In panel C, we've just quantified this difference in swarming rate between day 5 and day 8 with and without worms. So you can see that in the presence of E. coli, there is a response of M-xanthus to the worms, but in the F. johnsoniae patch, there is no such response. We hypothesized that this response happened between day 5 and day 8 because we initially added larval worms to the assay plates, and it took them about 5 days to mature and to start laying eggs and reproducing. So during this time, the worm population was actually growing, and we hypothesized that M-xanthus was responding to the increasing worm population size. So we did another assay to test whether the swarming rate of M-xanthus in each type of prey was dependent on the number of worms. We didn't find this was the case in E. coli, although we did see a response to the number of worms in F. johnsoniae patches. So in terms of the original response we saw of M-xanthus to the presence of worms in a patch of E. coli, there must be something else that is modulating this interaction between the two predators. We found some interesting things in this study. We found some things that we did expect, like that both C. elegans and M-xanthus could prey on both of the basal prey, and both of them preferred E. coli as a prey over F. johnsoniae. But we also found some things we didn't expect. We did not expect that C. elegans would tend to, in general, behave as if there was no food on plates that had M-xanthus only, in the sense that most of the worms we inoculated onto those plates ended up crawling up the sides of the plate and then dying in the search for a better source of food. We also didn't expect that on plates where we had all 4 of the organisms, it would be M-xanthus that seemed to dictate the regions of the plate where C. elegans was willing to go, rather than the other way around, that C. elegans would go to the best prey areas and M-xanthus would sort of take the areas that were left over. Because that's what we see in larger organisms. We see that apex predators just sort of do what they want. They find the best foraging areas, the best hunting areas, and then mesopredators kind of go along behind them in the areas that they've left alone in order to avoid the risk of being killed by the apex predator. But that's not what we saw in this system. So we don't see C. elegans behaving as an apex predator here. We see it behaving as if M-xanthus
either isn't a palatable food source for it or potentially is producing some kind of toxic compound or something like this, which is entirely possible because M. xanthus is known to have a large secondary metabolome, and a lot of these compounds we still don't know what they do. So there's definitely a lot more work that needs to be done for us to really understand the way these two predatory organisms interact and how they might interact in nature and how they might influence each other's behavior and each other's evolution in nature.
There's a paper by Franco et al. That came out in PNAS in 2019 where they look at how precipitation influences the microbial communities in different types of grasslands. What they found is that drought conditions have a stronger adverse effect on predatory nematodes than they do on root-feeding nematodes. So when there is a drought, there's actually a change in the functional composition of the soil community, with the predatory nematode population decreasing, and this allowing an increase in the population of root-feeding nematodes. Such a shift in functional composition has potential adverse effects, for example, for the plants that grow in these grasslands. Results like this have implications for how climate change will influence global food security, and it's one reason that studying microbial interactions is really important. I don't know what to do with my hands. Okay, I have to fix my eyes. Just keep going, just keep going.
Okay.